So my name is Ulrika Krus, and I work at the Lund University Diabetes Center in Sweden. I will tell you a bit about collection of human tissues and data. First, I will talk a bit about the facility that I'm working with. It's called the Human Tissue Lab, and it's a collaboration between the Swedish universities Uppsala and Lund. We are focused on diabetes treatment and research, and clinical human islet transplantation is the prime objective. That means that we get pancreases from deceased donors, isolate islets, and use them for transplanting the islets to type 1 diabetes patients that has a poor glucose control. We are also a world-leading human islet research facility where we collect samples in a biobank and also a database. HTL is divided into two parts. The clinical part in Uppsala deals with sample collection, islet isolation, QC for clinical transplantation, islet distributions to different centers and storage, whereas the research part in Lund is responsible for tissue distribution to researchers, nucleotide extraction, data generation, data QC and analysis, and data distribution. So what is a biobank sample? It is a biological material from humans that are not stored for more than 2 months. So that does not include samples taken for immediate diagnosis or treatment of the donor. The samples must be able to be traced back to the donor, and normally there is phenotypes available for the donor, for example, age, gender, and disease status that makes them useful for research. This is a flowchart of sample collection, and I will go through all the different steps for you. It starts with informed consent, followed by sampling, processing, adding the samples into a laboratory information management system, and then storage. So informed consent. It is of utmost importance since the consent sets the rules for future use of the samples. It's important that individuals are properly informed and has the capacity to make the decision to participate under no pressure. It's also important that the treatment of the patient will not be affected whether they choose to participate or not. A broad consent is preferred for biobanking, where they consent to storage and future research. But of course, there are so many different consent types that has to be considered. Consent can be withdrawn at any time, and samples must then be destroyed. There are some special cases where the donor is deceased. Then if there is a donation form filled in, then it's quite easy to follow that. But if not, there are country-specific rules about how to proceed. As we heard in Latvia, there were specific rules, and in Sweden we are very liberal, and we consider that if there is no donation form filled in and the relatives don't know what the will is, we presume that the donor is positive to donation. But there are different rules in different countries. For children, the consent is given by the parent or the guardian. But for older children, it is common that the child is present and gives the consent together with the parent. Sampling. I will go through some general considerations, liquid-based samples and solid tissues. Things you have to consider is, first of all, the stability of the sample. What kind of analysis will the sample be used for? There are different methods for different downstream analysis. You should always use standard operating procedures to ensure quality of the samples and reproducibility, because you do not want to include any technical variance in your storage. It's important with pre-label containers, especially if you're dealing with samples taken in the clinics where the sampler might be a physician who takes the samples during surgery, and then it's very important that the samples are put in a good container and make it clear where everything belongs. The temperature is important, as well as the time. For liquid-based samples, blood is the most common sample. It could be either anticoagulated, where you get plasma, buffer coat, and erythrocytes, or coagulated, where you get serum, blood, and blood clots. So this is a centrifuged blood sample, whole blood sample, which generates plasma, which is 55% of the total blood, buffer coat that contains leukocytes and platelets, and erythrocytes, which is red blood cells, which is 45% of the total blood. Blood is collected in color-coded vacuum tubes with different caps in different colors, where the red is for serum and purple is for whole blood. And then there is a whole lot of other containers with for different clinical measurements. And as I said, centrifugation of whole blood separates the phases. This is a summary of the different blood fractions and how you use them. So whole blood, you add anticoagulant and maybe also protease inhibitors. And this is a good source for RNA and DNA. The buffer coat, another part of the whole blood, is used for extraction of lymphocytes. Serum, where you add nothing, is used for proteomics and also for DNA, even though the yield is quite low. The same is true for plasma, where you add anticoagulant. You can also use it for proteomics and DNA. The blood clot, which is the remains when you centrifuge whole blood without anticoagulants, can be used for DNA extraction, even though it is quite costly and not is not the recommended one. Other liquids that are collected is urine. Normally the samples should be kept on ice or be refrigerated. Preservatives can be necessary, for example, EDTA. You can centrifuge the samples to get a cellular urine and a pellet, which can be stored separately. Condensed urine on filter paper is a good storage, specific effective storage method. Saliva is a good opportunity for self-collection of participants. They can then use swabs, cytobrushes, or mouthwash and send the samples in instead of meeting someone to sample them. This can generate good quality DNA and is suitable for large population studies. Solid tissue samples can be generated from different sources. For example, biopsies, leftovers from surgery, post-mortem samples, and transplants. What is important is that the sample should immediately be placed in a container with biopreservation media and be cooled to 8 degrees. Then there are several ways of biopreservation. Room temperature storage. That is used for formalin fixed paraffin embedded tissue blocks, which can be stored almost indefinitely at room temperature. These sections are mostly used for morphological studies. This is the oldest way of saving tissues. So if you want to use historical samples, they are normally in this format. Nowadays, it's possible to extract RNA and DNA from these tissue blocks, which is very good if you have an old sample collection which only contains this storage type. Filter paper card are normally used for blood and saliva, but there are also new methods which allows DNA, plasma, and serum. And it's used for extraction of DNA for genetic studies. It can be used for large cohorts since it's a very space-effective storage method. And normally you don't need very much DNA for these types of studies. Cryopreservation can be done in two different ways. Snap freezing, which is immediate lowering of temperature to -70 or lower. It can be done in liquid nitrogen, dry ice, or in a -80 freezer. It prevents the formation of water crystals and avoids degradation of nucleotides. Slow freezing is for preservation of viable cells, and then it's necessary to have a cooling rate of 1 degree per minute. That allows a flux of water from cells to avoid intracellular ice formation. Cryoprotectants, for example DMSO, circumvents cryoinjury caused by toxic changes in cell volume and solute concentrations and leaves the cells viable. DNA extraction is a very common thing to do with your biobank samples. If you have a large-scale cohort, the DNA is mainly from blood. And with large cohorts, a robot extraction method is recommended. You can also outsource the extraction to specific companies who specialize in these activities. It is, of course, also possible to extract the DNA manually. We are using a QIAGEN kit, but there are many other different kits for extraction of DNA. It's recommended when you do small-scale studies because for thousands of samples, manual extraction is very tedious. RNA extraction depends on the tissue. Different tissues have different obstacles, and you need specific methods for each tissue type. For example, homogenization is difficult in solid tissues like muscle, whereas degradation of RNA is problematic in liver, which contains lots of RNases. Adipose tissue, on the other hand, contains very few cells per weight and requires big volumes. When you have generated all your samples, it's very useful to have a system that keeps track of the samples. We use a laboratory information management system, which is an all-in-one place for sample information. All samples get a unique ID, which makes it easy to search and query. The traceability of the sample is very good, and you can see the history and origin of the sample. You also get an excellent overview of the storage locations. You keep track of the quantity and concentrations, and also the count of free store cycles. This is an overview of the LIMS that we are using. It's called LabVantage from SoftwarePoint. There are, of course, many other systems as well. As you can see here, there are different arms, so you can handle, there is sample management, storage, package, reports, and I have my own arm, which I have designed for myself to make it suit my needs. This is just an example of what it looks like. This is the storage of our DNA for 50 nanograms per microliter. So you get a list of all the samples, the quantity, and the location of the samples in the freezer. You can also add a lot of information of the sample if you have clinical data, analysis done, and so on. So this is a very convenient thing to use if you have a lot of samples. Some storage guidelines. Room temperature is used for slides, tissue blocks, and paper cards. whereas refrigerator is mostly for fresh samples in processing. Freezer up down to -27 is used for short-term DNA stability, whereas freezer up to -40 is for DNA stability. -40 to -80 is recommended for DNA and RNA, and -80 to -130 for urine and blood fractions. Liquid nitrogen vapor with a temperature of -130 to -150 is recommended for tissues, and liquid nitrogen, the liquid phase at -196 is recommended for living cells. Data generated from biobank samples are very often omics data. We have genetics and genomics, which origins from DNA. The genome contains about 40,000 genes, where about 20,000 genes are protein coding. Transcriptomics comes from RNA, where there are around 150,000 transcripts. Proteomics comes from protein, where the proteome consists of around 1 billion proteins, whereas metabolomics deals with metabolites, where there are around 3,000 compounds. We are working mostly with genomics and transcriptomics, and I will tell you a little bit about those methods. Genome-wide association studies is a method to identify common genetic variants, single nucleotide polymorphisms, that determine complex traits and diseases. It compares the distribution of genetic variants between groups. For example, is an allele more common in patients than in a healthy individual? Are individuals who carry allele G taller than allele A carriers? Normally, it includes 500,000 to 50 million variants in a GWAS. This is an example of a case-control GWAS, where we have cases with a BMI above 30 and controls with a BMI less than 20. And right now we are looking at this SNP, which has, in the normal population, the expected prevalence of the G allele is 36%, whereas there is supposed to be 64% of the A allele. But we observe in the cases that there are fewer with the G allele and more with the A allele. On the contrary, in the controls, it's the other way around, and the G allele is more increased and the A allele is decreased. So we do a statistical test and say that this difference is significant. And in the GWAS, you look at all the SNPs and do a statistical test for every SNP. And then, of course, you compensate for multiple testing, since you have maybe 500,000 tests. And then you find out which SNPs are different in the cases compared to the controls, and can continue with replication and biological studies. Transcriptomics. Transcriptome is the complete set of RNA transcripts that are produced by the genome under specific circumstances or in a specific cell using high throughput methods. A snapshot of gene expression at a given time in a given tissue. There are two different methods of measuring RNA expression. Microarray was more used before. It's where you use probes attached to a chip. Nowadays, it's more common to use RNA sequencing. In RNA sequencing, you extract mRNA from a tissue and convert it to cDNA. You sequence it on a high throughput sequencer, assemble and compare to a reference library to identify sequence variation, and counter reads to quantify expression levels. With this method, you get at the sequence and the expression levels and also the splicing patterns. Some example of usage of sequencing data is differential expression, for example, healthy versus disease, expression influenced by various treatments, pathway analysis, expression patterns in various tissues, expression related to other phenotypes. We mostly look at hormone secretion. You can look at many other different phenotypes like BMI, age, gender. EQTL, which is the expression related to a specific genotype. Single-cell sequencing is a new method where you look at the sequence in single cells. What I talked about before was bulk sequencing, which measures the average gene expression levels across a population of cells. That is useful for quantifying gene expression in homogeneous populations. It's cheaper, easier, and more straightforward to analyze, whereas single-cell sequencing measures the distribution of gene expression levels across a population of cells. It's useful for studying cell-specific transcriptomic differences in heterogeneous populations. It's more difficult to collect and analyze, but allows us to study new questions. This is a typical experimental single-cell workflow, where you start with your solid tissue, dissociate and generate single cells, extract RNA, convert it to cDNA, amplify the cDNA, attach it to a sequencing library sequence, and look at single cell expression profiles. And then you can identify different cell type specific expression signatures. Single cell sequencing is very useful for identifying novel and rare cell types, understanding heterogeneous tissues, looking at changes in cellular composition, trace lineage and developmental relationships between cell states. This is an example of combining omics-data that is from our center, where we looked at the melatonin receptor. And we saw in GWAS studies that the SNP, this melatonin receptor SNP, had an effect on insulin response to glucose in a big cohort with 3,300 individuals. So you can see that the insulin response is decreased with the genotype. So the CC allele has the highest effect. The heterozygous has a decreased effect, and then the homozygous GG has the lowest response. We then looked at RNA sequencing of the melatonin receptor and could see that the GG carriers had a higher expression of this receptor. When looking at insulin secretion, we saw that adding melatonin decreased the secretion of insulin. So this is a way of combining GWAS with expression studies and then following up with functional studies showing that melatonin has a negative effect on insulin secretion. When dealing with human data, it's important with regulations. GDPR, the General Data Protection Regulation, is a European regulation that was started 25th of May 2018. And this regulation overrules all national regulations. The purpose of GDPR is to harmonize data privacy laws across Europe, to protect and empower all EU citizens' data privacy, and to encourage the lawful free flow of data across Europe. What kind of data is affected by GDPR? It's almost all human data. Personal data, which is data relating to a natural person who can be identified by a reference to an identifier such as name, identification number, location data, and an online identifier. Sensitive data is specific data which deals with ethnic origin, health data, sexual life, genetic data, biometric data, political opinion, religious beliefs, trade union memberships, and criminal offenses. And for scientists, the first ones are the ones that we are mostly dealing with. Pseudonymized data is personal data that can no longer be attributed to a specific data subject without the use of additional information. And this additional information must be kept separately. And this is the way that we normally handle data. It's very unusual that scientists deal with personal information. It's almost always pseudonymized, even though it still applies, GDPR still applies. And also GDPR applies to anonymized data, which is information that cannot be linked to an identifiable individual. The general principles of GDPR are 7 statements. Lawfulness, fairness, and transparency. Reasons for using the data that applies to scientists is public interest and legitimate interest. It is important here that the data subjects has consented. You need to consider the effect on the data subjects when using the data, and you have to inform the subject on the data life cycle, how long the data will be stored, what it will be used for, and who is going to process it. Second is purpose limitation. Personal data can only be collected and processed for specific purposes. Data minimization means that you should only use necessary data. Do not process unnecessary data or data that could be avoided. Accuracy is to ensure that the data is accurate and up to date. Storage limitation. Data should be kept identifiable only for as long as necessary. Integrity and confidentiality. Protect the data against unauthorized and unlawful processing. And accountability. Document procedures to comply with data protection requirements. And these regulations are very important, and there are huge fines if you do not follow these rules. So this is something that you have to be very careful with when dealing with human data. Data repositories is it's almost all scientific journals require dataset upload if you're going to publish. And the sensitive data, like genetic data, must be protected against unwanted disclosure. The access to sensitive data must be safeguarded, and protection of sensitive data can be required for both legal and ethical reasons. So therefore, there are recommended repositories for sensitive data, which limits the access of data. There are 3 different archives, one on each continent. dbGaP, the database of genotype and phenotype, is the American one. It's sponsored by NIH, National Institute of Health. Their data access is restricted to researchers who request datasets for specific research uses, for institutional signing officials from the PI's home organization who certify and submit such requests, and for NIH staff who review and process requests. EGI is the European Genome Archive. It contains data collected from individuals whose consent agreements authorize data release only for specific research use research use or to bona fide researchers. Data access decisions will be made by the appropriate data access granting organization. This organization will normally be the same organization that approved and monitored the initial study. Then there is also a Japanese archive, Japanese genotype phenotype archive, which is very similar to the European one. It is provided in collaboration with the National Bioscience Database Center of Japan Science and Technology Agency, and the users apply for data access directly to the NBDC. So, to summarize, biobank samples can be traced back to the donors. Informed consent is essential for usage of biobank samples. Plan for downstream analysis when sampling. Storage temperature varies for different types of samples. Omics are used for research tools that provide lots of information. GDPR protects EU citizens' data privacy, and sensitive data such as genetic information needs extra protection. Here are some further readings if you're interested. You will get this presentation, so you don't need to write this down. Thank you.
Okay, thank you. Thank you. So I have a question. Is always like reality the way how you described, like according to all the standards? Or your cooperation with Uppsala is very well established and it's always like the standards?
Well, we of course try to follow the standards as much as possible. I mean, we're always following the standard operating procedures for all sampling, and of course we always follow the informed consent and only use samples where the informed consent applies. Sometimes the donors only accept to donate to transplantation, but not to research, for example.
Okay. And another question was about this living cells. So, like the standard temperature for living cells storage is -196 degrees. Is it correct?
Yeah.
Wow. And how you transport between like Uppsala and Lund?
We don't actually.
Okay.
This is done. This is done in one site, so we don't transplant those. We don't transfer those samples. We actually only transfer living islets and fresh samples on ice. So that's how we transfer. The other things are done at one site only. So most of the storage is done in Uppsala.
Ah, okay, okay, thank you. But like this islets transplantation, maybe you mentioned, but I didn't hear. But it's now done in mice, yes?
No, this is done.
You also do it.
This is in human, yes.
Okay, and is it successful?
Yes, it is. It's quite successful. If you transplant islets you could. It's not like normally you can't be totally off insulin. So normally you need to take some insulin as well, but your insulin levels gets more stable and the blood glucose is more stable. The problem, but there are of course also those who can leave insulin totally. It's not permanent. It's the islet survives for around 5 years. Then you need to do a new transplant.
Okay, thank you. Very interesting. Okay, maybe someone else has some questions. Okay, it looks that no. Then thank you very much, Ulrika. It was very valuable. Like we don't have in our biobank such a big experience with tissue data, but we would of course like to develop this and hopefully in future. So yeah, it is a very nice introduction for us. Thank you. Okay, to everyone else who is here, I will share my screen.